So hello, this is the second in the series of the PDBe's API webinar series, and this one will be about searching the PDBe with our API. I am John Beresford, one of the curators at PDBe, and I'll be walking you through this webinar of what features we have in our search. As I mentioned, this is the second in the series. There are another 4 presentations in the following weeks after this. Each one of these has a separate sign up, and I do encourage you to sign up for any of the other webinars that may be of interest to you. So in this webinar, I will go through how we sit as part of the PDBe as PDBe, our search system, how it works, our advanced search, introduction to our API, and then go through some usage examples to show how it works and what you can do with our search. So PDBe is part of the PDBe. We sit here in the UK. Together with RCSB, PDBJ, and BMRB, we form the PDBe. EMDB is likely to join the PDBe at some point in the near future, and together all 4 of us produce the archives, which is the EMDB archive, the PDBe archive, and the BMRB archives. And you can get the same archive from any of the sites that are mentioned here. During deposition, which is commonly used in the one-dep system, which is deployed at PDBe, RCSB, and PDBJ, we get data provided by depositors, and we add extra data during curation. This includes data about experimental methods, sequence information, connectivity validation, and we produce the archive MMSIF and validation XML files. And as I mentioned, this is available from the FTP site of every of the sites there. And then we do things a little bit differently depending on which site we are. So for PDBe specifically, we are down here. We are at the heart of EBI's resources. So we interact and we receive data from, and we share data with lots of the services mentioned here, which form part of the EBI infrastructure. This allows us to add additional data to the data that you get just from the MMSIF file. An example of this is the SIFTs project, which is mentioned here, which is a collaboration with Uniprot, where we add additional data from gene ontology, Interpro, Uniprot, PFAM, and structural domains of SCOP and CATH all together into our data that we have and we serve directly from PDBe. So then we prepare all this data and allow you to search through it. So why search the PDBe? What would you be looking for? So it provides a nice convenient way of finding individual entries that you'll be looking for in EBI, in the PDBe. So for example, you might want to find all of the hemoglobins solved by electron microscopy, which you can do directly in our search. Which proteins bind to paracetamol, again available directly through the search, and which ligands bind to hemoglobin apart from just heme. So how would you go about doing all of these things? So our search is available directly from our homepage, which is pdbe.org, and it's highlighted here in red on the top right-hand corner. It features an autocomplete, where if you start typing, you get results in different categories, which allow you to narrow down your search into exactly what you want. So here I've started typing hem, and that could be matching an author of hemming, a journal of hematology, the ligand heme, or hemagglutinin, and you can select which ones of these you want. If you want to do more advanced searching, you can use our advanced search functionality, which is available again in the same place on the home page, but just underneath the search button, there is a button here with advanced search, which allows you to build more complex queries. So it contains the same autocomplete functionality as you start typing. Like for example, I started typing homo here, and we can see all the things that have matched that in the organism name here. And then you can build up additional search parameters. So here I've done Homo sapiens and hemoglobin subunit alpha. But we can change these ands into different operators. So we can and or not ignore. And we can change the regular expression from contains equals starts with ends to allow us to build more complicated queries. So on the left here, we have, for example, hemoglobin subunit alpha, not from human, and that would get us that. And on the right here, we have the hemoglobin subunit alpha from either human or sperm whale, so allow us to put an or into here. While using this advanced search as well, we can use wildcard expressions. So we can search using question marks and asterisks within the search. For example, we could substitute a single letter with a question mark. So this particular one here would match both test and text. And we can do a star and asterisk, where we could do test would match test, testing, tester, where it matches multiple characters. After we've done the search and we've hit search, we get to see the results on the search page. And then in the top left, we can change the search by either clicking on these parts here or the back onto the advanced search. And then we get the results presented here in panels in the middle. And additional filters can be easily added using the facets. So here we've selected electron microscopy and added it, and this takes us to here where it adds the query directly into our search. So then what data do we see in the search? What do we get in the searches window afterwards? We get everything you can see within the search results page is here. For example, you can see the PDBe IDs, the data release, the experimental methods, taxonomy, source information, experimental details. We have crystallographic cell parameters, the citation that's available, the assembly, chemical components. So quite a lot of data is there. And we'll get into more of that as we go into using the API and using examples in there. An additional functionality we have within the advanced search is protein sequence searchings against the PDB. This can be done either using a FASTA search or a p-hammer search. So here in the advanced form, we've just typed in sequence search, and then we can pick these two here. And then when we search, we see a slightly different search results box. We get additional information here of the e-value, the target, including the chain ID, the percentage identity, and the alignment that we get back here. So as this is all about the API, we'll start introducing how we access all of these things through the API. So our search is powered by Solr, by Apache Solr. So we have indexed 214 data items. About half of them are available through the advanced search, 3/4 of those or so through the autocomplete functionality on the front page that I showed you, and only about 30 to 40 through the facets down the left-hand side. So there are a lot more data items available that are indexed that are not necessarily accessible through any of the UI search that we have. So in common with all of PDBe's web pages, all of our data is served through APIs, and they are directly accessible for the public to use. And search is no exception to this. So the landing page for our APIs, pdbe.org/API, lists the 3 API types that we have, each of which contains multiple calls and multiple things you can do. So the first one here that I mentioned is the Solr-based query system. Then we also have an entry-based API, which allows you to provide information about the entries, about an entry, or that being a PDBe entry or a ligand that we have. And then we have an aggregated API that allows you to provide aggregated data from several different entries all in one place. And these two, bottom two, will be the subject of later webinars. So we're going to focus on the Solr-based API that we have. So if we click on that link that was there, we get taken to the documentation page for the Solr search that we have. And it provides a list of the fields that are available for querying. For example, PDBe ID here, and then PDBe entry code, which is what's shown here in the red box. So the field name is the part before the colon, and then we have a text description of what is returned when you use that field name, so what data is in there. At the bottom of this page, we also provide a interactive query tool, which allows you to try your own queries directly on the page and run them on there. So it's here on this thing, we can see this data here, and then if we hit run call, we can get back the URL that was used to make the call, and the results are shown just below here. So we can change the search directly in this query box. So below in the docs section of this call here, and in the documentation bit, so any of the fields that before the colon or are in red in the docs section can be used in search. And here, for example, we have changed this query to all assembly type tetramer, and we're going to return all the assemblies that are tetramers. So before we get into actually looking at the results, I need to go through exactly how do we index data within our search. So as I mentioned, this is based on Solr, and a document is our unit of search and index. So within each document, there are fields, which is the properties of that document. So if we compared this to a database, a document would be a row in a table, and the fields would be the columns that correspond there. So one of the questions we had when designing this is for PDBe entries, what is the optimal document that we can have? So if we look at the hierarchy of a PDBe entry, we have on the left, starting with what's in the contents of the file that we receive. So in this case, it's 2 copies of this complex here. During annotation, we define this as the quaternary structure, or the likely quaternary structure of the entry. Within that quaternary structure, we know what are the unique polymeric molecules. And then within that quaternary structure, again, we can see what the individual chains are. And you can see here, there'll be 2 copies of each of these 2 chains in this one. Then we go down to residue levels to find out the interactions, and finally down to atom levels. So for search, we would have to devise a document that would allow us to find what users seem to search for. So what are all the molecules in entry, the properties of their molecules, so for example, the sequence, properties of the entry, such as quaternary structure, but also not create too large an index that the slow is full of additional data and returns unnecessary documents, so a lot of duplication. So at PDBe, we decided that the best part we could have is the unique polymeric molecules here. And the reason that we picked for doing this is because we can give you properties of individual molecules within this. So we can tell you the type of a molecule in an entry, the sequence of that molecule, the number of copies of that molecule with an entry and the associated chain IDs. If the sequence of that molecule has changes, which ligands interact with that molecule, not just are in the entry, which ones interact with that molecule. We can also say which other polymers interact with that molecule. So we can see in the example we've got here that the protein interacts with the DNA. So it's not just an entry that contains protein and DNA. We know that the protein interacts with the DNA, and we can highlight that within the search. So then how does this translate into doing search? So if we take a PDBe entry, for example, given here, a PDBe entry of hemoglobin, which contains 2 unique molecules, macromolecules. So that's 2 unique polymers that we have. We can see that if we run the query, we get 2 documents back. So you have to sort of do a bit of juggling around if you're just looking for PDBe entries, and I'll go through how to do that. But otherwise, you have a lot more data available about each molecule within the document. So now we'll run into some usage examples of how to use our API and what do we have in there. So as I mentioned, it is a Solr search, and it is directly accessible via a URL. And there are various parts to this URL that start to build up the query. So here we have our main search URL, which is this part here. Our query, which is denoted by Q, and then has our query field, and then colon, the actual value that we're searching for. And in this example, we have return type, which is the WT here. And the options are JSON and XML, and it defaults to JSON. So in subsequent examples, I won't show these defaults. And by default, it shows 100 documents, and we can change this by changing the rows parameter from 100 to a much larger number if we want. Because this is plain Solr, details of the exact syntax can be found in the Solr help pages. So we support both get and post calls, and in the examples here I'll sort of show a hybrid, so I won't show the full get query, which is an example shown here with the ampersands separating each of the parameters. What we can do is instead is a post where we post the data as JSON with key value pairs. So here we have exactly the same query, which I'll go through what these mean. FL, Q, rows, and WT are the keys, and then the values are shown here. So what do we get when we use this search? So by default, when we run a search, we get every single field within a document returned to us. So this is very, very useful when you're not quite sure what you're going to get and you want to just test queries out and you want to see how you join things together. One of the most useful things we have if you want to speed up your queries and you know what you're looking for is the FL, so the field list parameter here, which allows you to only return very specific fields back. And this will make a massive difference to the data content that you get back, and it'd be a lot quicker doing much larger queries. So here we've done our query of all assembly type being tetramer, and our field list, we're only going to get back PDBe ID and experimental method, which is a comma separated list. And as we can see in our response on the right hand side, that's exactly what we've got within our documents. We've got back experimental method and the PDBe ID. There are quite a few exclusive searches which only work currently within the search API, and these are a bit more advanced than you can use within the advanced search, in fact. So for example, it is possible to look for prosite patterns within sequences. So I've given a few examples here, looking for a phosphorylation site, and we'd query the molecule sequence with this prosite pattern we have here, a tyrosine kinase phosphorylation site, and we can do something similar. There are a few differences between the prosite syntax on the prosite website and what we use here. Very simply, we have changed the rules into standard regular expression rules for this to work. So if we're looking for any 2 or 3 residues rather than a prosite X 2 or 3, it becomes dot, and then curly braces 2 or 3. And not is a hat, as in a regular expression, and any of remains as standard square braces that we have. Another exclusive search we have that's currently not available anywhere else is looking for antibodies. So we run a program behind the scenes to identify antibodies with PDBe. The reason we do this is if you currently do a text search for antibody, you get very messy data back, and you get about 3,400 PDBe entries at the time I wrote this. What we do have within the search API, though, is a very simple flag of antibody Y for yes. So we can find all the entries that contain all the molecules that are antibodies, but maybe we want to find a number of PDBe IDs that we have that are containing antibodies. So if you think about an antibody structure within its individual PDBe entry, you might get multiple copies of an antibody in there. So multiple proteins that together form an antibody, and we want to group these together. So we can do this using, again, the same query, antibody flag Y, and then we can group these things, these results, by a particular field. In this case, we're going to group by PDBe ID. We're only going to turn groups when there is actually one, and the number of groups is, we're going to return number of groups that we have here. And I'm only going to return PDBe ID using a filter list like we had before. And then within the response that we get back, we can see that we have an N groups field that has appeared. And we've got 4,931 PDBe entries that likely contain an antibody, which is significantly more than we get from doing a free text search. Going through another worked example of the faceting that I showed at the very beginning of how this works within our search. So that, for example, we've searched for lysozyme here, and then we want to find out what are the different experimental methods that are used for solving the structures of lysozyme, and this is available in the facets on the left-hand side of our search results, which are shown here. This is possible directly in the search API as well, with a slight modification to the query that we just had for the grouping before. So we've done the same search we do in our page, where all molecule names is equal to lysozyme. We group our entries by PDBe ID, and then we're going to facet the entries. So we're going to facet as true, only things that actually come up with an answer we want to facet, and we're going to facet by experimental methods, and we're going to group our entry PDBe IDs into the facets as well. And on the right hand side, we can see what we get back. So this is the same numbers that we have within the actual search page in the main UI. We can also do something similar here using pivoting to do a very similar query. And in this case, we've just added facet pivot, and we will pivot on experimental method. And this gives us a slightly different representation, but containing exactly the same data. So we can see we now are pivoting on experimental method, and this becomes our field as well here. And we can see what the value and the count of entries in this pivot is. So it's just two methods of doing something similar. However, pivoting allows us an extra. It allows us to do multiple pivots to find multiple properties of things. So, and a more complicated example here, if we wanted to find the number of components in an assembly per experimental method in entries solved between 2014 and 2020, and we wanted to group the results by year, we can do this with a relatively simple query. So again, we're grouping these things together, we're putting into facets, and we're pivoting, but we're pivoting this time on a comma separated list. So we have three fields that we are pivoting on: release year, experimental method, and assembly number components. And we're just going to sort these things by release year as well to allow us to see them, although probably not necessary if you're handling this computationally. And if you can see in the results that we have here, we then have 3 levels of pivots. So starting with the first one was our full list. And then first of all, we pivot by release year, and then experimental method, and then by number of components that we have. So the results that we have, for example, here is that we only, if entries that have only 1 component, there are only 4 done by electron microscopy in 2014. And it allows us to get that granularity of our results. We have provided and made available tutorials of how to use a lot of these features on our GitHub training pages, which are shown here, which provide Python walkthroughs of how to get data here. And the specific example that we have for searching is available in part 6 of this series. I mentioned earlier that we have a sequence search available, and this of course is also available through our API. So both the FastA and pHammer searches are available. Handling the results is a little bit more complicated than I want to go through in this webinar, but it is covered in lots of detail in the 7th part of the API tutorials that I have shown here, which will walk through how to use this feature. So we're drawing to the end of the webinar now, and just to summarize, so PDBe provides an advanced search which is powered by Solr. It is one of our 3 distinct APIs, that being search, an entry-based API, and an aggregated API. Like the others, the search system is accessible directly via the API. Exactly everything you see on the web pages can be accessed programmatically. This includes quite complex queries and some advanced queries that we have. And this includes the FastA and Hammer searches that I showed. The tutorials that we have are available through the GitHub page of how to use this with some Python walkthroughs. So that's coming to the end of this webinar. The next webinar that we have will be Neural. My colleague here will be presenting how to create complex queries using our entry API. And this is a separate sign up from this webinar. So you'll have to visit one of the following URLs. So remember, you have to register for each webinar individually. And I would like to thank you for your attention and ask if there are any questions that you would like to ask.